Good morning, students. Today we have lecture number 3 for histology, and our theme today is the cardiovascular system. And we begin with overview of the cardiovascular system. The cardiovascular system is concerned with the transport of blood and lymph through the body. And the cardiovascular system consists of the blood and lymphatic vascular system. And you can see that cardiovascular system can be divided on blood vascular system and lymphatic vascular system. And function is transport of blood and lymph through the body. Let's look to structures of cardiovascular system. The blood vascular system is composed of the following structures. First, the heart. And function of the heart is to pump the blood. Pump the blood. Next structures are the arteries. The arteries. And their function is to carry blood with nutrients and oxygen to the tissues. To carry the blood with nutrients and oxygen to the tissues. So the direction to the tissues. The next structures are the capillaries. The capillaries. Capillaries are diffuse network of thin tubules through whose walls the change between blood and tissues takes place. So this is a diffuse network of thin tubules. And function of capillaries is to change between blood and tissues. And the last structures are veins. The veins. Function of veins is to convey products of metabolism toward the heart. To convey products of metabolism toward the heart. So it's in other direction, from tissues to heart. And lymphatic vascular system. Lymphatic vascular system begins in the lymphatic capillaries. Begins in the lymphatic capillaries, which are blind-ended tubules. Blind-ended tubules that terminate in the blood vascular system, emptying into the large veins near the heart. And the function of the lymphatic system is to return to the blood the fluid of the tissue spaces. The function is to return to the blood the fluid of the tissue spaces. And you can see that lymphatic capillaries begin like blind-ending tubules and emptying into the large veins near heart. Let's look to general structure of blood vessels. The general structure of blood vessels. The blood vessels have a common basic structure. Blood vessels are usually composed of the following layers or tunics. So all blood vessels are usually composed of the following layers or tunics. The inner tunic. First, Tunica Intima. Tunica Intima. Tunica Intima consists of, first, the layer of endothelial cells. Layer of endothelial cells lining the vessel's interior surface. Lining the vessel's interior surface. These cells rest on a basal lamina. These cells rest on a basal lamina. Next layer, subendothelial layer. Subendothelial layer consists of loose connective tissue. Consists of loose connective tissue. And in arteries, the intima is separated from the media by an internal elastic lamina. In arteries, the intima is separated from media by an internal elastic lamina, which composed of elastin fibers. Composed of elastic fibers. These three layers of Tunica Intima. Next, Tunica Media. Tunica Media. Tunica Media consists of smooth muscle cells. Tunica Media consists of smooth muscle cells. And smooth muscle cells and elastic and reticular fibers. The smooth muscle cells and variable amounts of elastic and reticular fibers. In larger arteries, a thinner external elastic lamina separated the media from the outer Tunica Adventitia. In larger arteries, thinner external elastic lamina separated the media from the outer Tunica Adventitia. And the outer layer, external layer, is Tunica Adventitia. Tunica Adventitia consists of loose connective tissue with longitudinally oriented collagen and elastic fibers. So Tunica Adventitia consists of loose connective tissue and longitudinally oriented collagen and elastic fibers. And it contains nerves and blood vessels which have name vasa vasorum, Vasa vasorum, blood vessels of blood vessels. And blood vessels are structurally adapted according to hemodynamic factors. All blood vessels structurally adapted according to hemodynamic factors. So here you can see scheme of the general structure and histological microslide with artery structure. Look, the scheme. Tunica Intima consists of endothelial layer, is simple squamous epithelium. Subendothelial layer, loose connective tissue with longitudinally oriented elastic and collagen fibers, and internal elastic lamina in large arteries. Internal elastic lamina. Medium layer. Tunica Media. Tunica Media consists of smooth muscle cells and some fibers, elastic and reticular fibers. And tunica externa, outer tunica, is Tunica Adventitia. Tunica Adventitia consists of loose connective tissue with blood vessels and nerves. And you can see between Tunica Adventitia and Tunica Media external elastic lamina, which we can find in large arteries only. And microslide, image of microslide of the blood vessel, artery. You can see Tunica Intima consists of endothelial layer, very thin subendothelial layer, and we can find elastic internal elastic lamina. Very thick layer of Tunica Media, smooth muscle cells, and Tunica Adventitia. Let's look to arteries. Arteries transport blood to tissues. Arteries transport blood to tissues. They resist changes in blood pressure and in their initial portions and regulate blood flow in their terminal portions. They resist changes in blood pressure in their initial portions and regulate blood flow in their terminal portions. And there are three main types of arteries. Three main types of arteries. First type, elastic or large arteries. Elastic or large arteries. Second type, muscular or medium arteries. Muscular or medium arteries. And small arteries or arterioles. Small arteries or arterioles. Elastic arteries. Elastic arteries. The blood is pumped from the heart into large elastic or conduction arteries. Blood is pumped from the heart into large elastic conduction arteries. The aorta and its large branches are the typical elastic arteries. the aorta, and its large branches are the typical elastic arteries. And elastic arteries have the following characteristics. Elastic arteries have the following characteristics. First, Tunica Intima. Tunica Intima consists of 3 layers. Inner one, endothelium. Endothelium is smooth without of folds. Endothelium smooth without of folds. Second layer, subendothelial layer. Subendothelial layer is thick and contains collagen and elastic fibers. Thick and contains collagen and elastic fibers. And third layer is internal lamina. Internal lamina, which is present here. So here you can see on the slide Tunica Intima, Tunica Intima, endothelial layer, subendothelial layer, and internal elastic lamina. Internal elastic lamina. Next, Tunica Media. Tunica Media consists of concentrically arranged perforated elastic laminae. Tunica Media consists of concentrically arranged perforated elastic lamina. You can see Tunica Media in aorta is very thick and contains of perforated elastic lamina. And we can see a few amount of smooth muscle cells between layers of elastic lamina. And Tunica Adventitia. Tunica Adventitia consists of loose connective tissue, contains elastic and collagen fibers, blood vessels and nerves. Tunica Adventitia consists of loose connective tissue, contains elastic and collagen fibers, blood vessels and nerves. Muscular artery. Muscular arteries. Blood passes from the elastic arteries via arteries of intermediate type into the muscular distribution arteries. Blood passes from the elastic arteries via arteries of intermediate type into the muscular distribution arteries. And most of the arteries in the human body are muscular arteries. Most of the arteries in the human body are muscular arteries. In muscular arteries, the blood passes at a reduced pressure and speed. In muscular arteries, blood passes at a reduced pressure and speed. The size of their lumen is controlled by a contraction or relaxation of smooth muscle on their wall. The size of their lumen is controlled by contraction or relaxation of smooth muscle on their wall. Muscular arteries have the following characteristics. Muscular arteries have the following characteristics. First tunica is Tunica Intima. Tunica Intima forms numerous small folds. You can see Tunica Intima forms numerous small folds. And Tunica Intima consists of endothelium is simple squamous epithelium, subendothelial layer, contains a few smooth muscle cells, and internal elastic lamina. Internal elastic lamina is prominent and forms small wavy folds. Tunica media. Look, this tunica is very thick. Tunica media comprises a thick layer of circularly arranged smooth muscle. External elastic lamina is present in large muscular arteries. External elastic lamina is present in large muscular arteries. And Tunica Adventitia. Tunica Adventitia consists of loose connective tissue, contains elastic and collagen fibers, and blood vessels, vasa vasorum, and nervous tube in Tunica Adventitia. Arterioles, the smallest arterioles. These are generally less than 0.5 millimeters, less than half of millimeter in diameter, and have relatively narrow lumens. The wall of the arterioles contains Tunica Intima forms numerous small folds. Tunica Intima forms numerous small folds, consist of endothelium is simple squamous epithelium, subendothelial layer is very thin here, and internal elastic lamina is prominent and forms small wavy folds. Tunica Intima consists of endothelium, simple squamous epithelium, subendothelial layer, very thin, and internal elastic lamina is prominent and forms small wavy folds. Tunica media of arterioles composed of one or two circularly arranged layers of smooth muscle cells, composed of two or one circularly arranged layers of smooth muscle cells. External elastic lamina is absent. External elastic lamina is absent. And Tunica Adventitia. Tunica Adventitia consists of loose connective tissue. Arterioles are very thin with very narrow lumen. On this slide we can't distinguish clearly all these layers, but we can see media part, nucleus of smooth muscle cells, and the blood inside the lumen. Veins. The veins return blood to the heart, aided by the action of smooth muscle and specialized valves. Veins return blood to the heart, aided by the action of smooth muscle cells and specialized valves. And here you can see classification of the veins. Classification of the veins. There are 3 types of veins: unmuscular, atypical, muscular, and venules. Unmuscular, atypical, muscular. Muscular can be subdivided on 3 types. Muscular veins with weak development of muscular elements. Muscular veins with weak development of muscular elements. Muscular veins with middle development of muscular elements. With middle development of muscular elements. And muscular veins with strong development of muscular elements. There are 3 types of muscular veins, with weak, middle, and strong development of muscular elements. And the smallest one are venules. Venules. Begin with unmuscular veins. The unmuscular veins are in organs with dense walls. For example, meninges, bones, spleen, with which they strongly fuse external tunic. The unmuscular veins are in organs with dense walls, like meninges, bones, spleen, with which they strongly fuse external tunic. The wall of these veins consists of endothelium, which is surrounded by a layer of connective tissue. Smooth muscle cells are absent. They have very thin wall, which consists of endothelium, which is surrounded by a layer of a connective tissue. And smooth muscle cells are absent here. Muscular veins. Muscular veins. You can see muscular veins with weak development of muscular elements, with middle development of muscular elements, and with strong development of the muscular elements. Muscular veins with weak development of muscular elements. These veins are above the level of the heart, on which blood goes passively owing to weight. These veins, muscular veins with weak development of muscular elements, are above the level of the heart, on which blood goes passively owing to weight. The structure of the wall of these vessels has the following characteristics. Structure of the wall of these vessels has the following characteristics. Tunica Intima. Tunica Intima consists of endothelium, subendothelial layer is weak developed, subendothelial layer is weak developed, and internal lamina is absent. So only two layers here. Tunica Intima, number 1, consists of endothelial layer and weak developed subendothelial layer. Internal lamina is absent. Tunica Media is thin with small amount of smooth muscle cells. Tunica Media is thin with small amounts of smooth muscle cells. External elastic lamina absent. External elastic lamina is absent. And Tunica Adventitia. Tunica Adventitia consists of loose connective tissue with blood vessels and nerves. Muscular veins with middle development of muscular elements. These veins are on the level of the heart. These veins are on the level of the heart. The structure of wall of these vessels has the following characteristics. Structure of the wall of these vessels has following characteristics. First, Tunica Intima. Tunica Intima forms the valves and consists of first endothelium, number 1. Subendothelial layer is weak developed. Subendothelial layer is weak developed. Internal lamina is absent. Internal elastic lamina is absent. Tunica Media. Tunica Media consists of a few layers of smooth muscle cells. Consists of a few layers of smooth muscle cells. External elastic lamina is absent. External elastic lamina is absent. You can see number 2, Tunica Media, middle development, and consists smooth muscle cells without external elastic lamina. And Tunica Adventitia, number 3, Tunica Adventitia, consists of loose connective tissue. With blood vessels and nerves. And muscular veins with strong development of muscular elements. You can see strong development of muscular elements. These vessels are below the level of the heart. Below the level of the heart. And these veins contain well-developed bundles of smooth muscle cells in all three tunics. These veins contain well-developed bundles of smooth muscle cells in all three tunics. In Intima and Adventitia, bundles have a longitudinal direction. On the media, they have circular direction. We can find bundles of smooth muscle cells in all tunics. In Intima and Adventitia, longitudinal direction. In media, circular direction. There are numerous valves. Valves consist of two semilunar folds of the Tunica Intima that project into the lumen. You can see folds that project into the lumen. They are composed of elastic connective tissue and are lined on both sides of endothelium and lined off on both sides by endothelium. And you can see endothelial layer, subendothelial layer, Tunica Media, very thick layer of Tunica Media, and Tunica Adventitia. All this layer contains smooth muscle cells bundles. And the smallest structures, venules. The venules have very thin walls. They have very thin walls. Tunica Intima consists of endothelial cells. Tunica Intima consists of endothelial cells. Tunica Media may contain only contractile pericytes, but a few smooth muscle cells are usually present. But they can contain only contractile pericytes. And Tunica Adventitia consists of loose connective tissue. And here you can see the venule. This is the venule, and upper the arteriole. The venule. Tunica Intima consists of endothelial cells. Tunica Media contains pericytes, and Adventitia consists of loose connective tissue. Capillaries. The capillaries. The capillaries are microscopic vessels with diameter about 8 picometers. Capillaries are microscopic vessels with diameter about 8 picometers. They branch and anastomose to form a diffuse network. They branch and anastomose to form a diffuse network. Capillaries have structure to permit metabolic exchange between blood and surrounding tissues. Capillaries have structure to permit metabolic exchange between blood and surrounding tissues. General structure of capillaries. The general structure of capillaries. Capillaries are composed of. Capillaries are composed of. First, the single layer of polygonal endothelial cells. Single layer of polygonal endothelial cells. External surface of endothelial cells rests on the basal lamina. External surface of endothelial cells rests on the basal lamina. Second layer, medial layer. Pericytes contains of pericytes are cells with long cytoplasmic processes. Cells with long cytoplasmic processes that partly surround the endothelial cells. That partly surrounds the endothelial cells. Pericytes have contractile function. Pericytes have contractile function. And the adventitial cells. Adventitial cells is stem cells. Adventitial cells is stem cells. On first picture, you can see the diffuse network of capillaries. This is capillaries network. Very thin lumen which can contain only one row of red blood cells, erythrocytes, or sometimes two rows. And on the second picture we can see the capillary with endothelial cells and red blood cells in them. And capillaries have special classification according of structure of their walls. Classification of capillaries. There are three main types of capillaries. There are three main types of capillaries. The first one is continuous or somatic. Continuous or somatic. These capillaries have uninterrupted lining of endothelial cells and basal lamina. This type of capillaries have uninterrupted lining of endothelial cells and basal lamina. Distribution in human body. Distribution in the human body. All kinds of muscle tissue, in connective tissue, in exocrine glands, and in nervous tissue too. Distribution in the human body. All kinds of muscle tissue, connective tissue, exocrine glands, and nervous tissue. Second type of capillaries is fenestrated or visceral. Fenestrated or visceral. This type have large fenestra that are closed by a diaphragm. These type of capillaries have large fenestra that are closed by a diaphragm. A continuous basal lamina is present. Basal lamina is continuous. And distribution of these capillaries. For example, in endocrine glands, and glomerulus of kidney. Glomerulus of kidney. And third, the last type of capillaries is discontinuous sinusoidal capillaries. Discontinuous sinusoidal capillaries. These capillaries have wide diameter, about 30-40 micrometers. They have wide diameter, 30 to 40 micrometers. The endothelial wall is discontinuous, and endothelial cells show multiply fenestrations without diaphragms. Endothelial cells have multiply fenestrations without diaphragms. And the basal lamina is discontinuous too. The basal lamina is discontinuous too. Distribution in human body, in the liver, hematopoietic organs, for example bone marrow, red bone marrow, and the spleen. Discontinuous sinusoidal capillaries. Distribution, liver, red bone marrow, spleen. On the next picture, you can see the scheme or structure of three types capillaries. First one, continuous. Continuous or somatic. And you can see endothelial cells uninterrupted, and they have uninterrupted basal lamina or basement membrane. Uninterrupted endothelial cell, basement membrane, and pericyte. The second type, fenestrated capillaries. fenestrated. In endothelial cells we can see a numerous amount of fenestra, special pores, and you can see that endothelial cells has fenestra, but these fenestra closed by diaphragm and basement membrane, still uninterrupted, and we can see parasite here. And sinusoid capillaries. Sinusoid capillaries have pores. Sinusoidal capillaries have pores. They have multiple fenestration and have no diaphragm in this place. And you can see that basement membrane interrupted too have pore. Then we look to lymphatic vascular system. Lymphatic vascular system. The lymphatic vascular system consists of endothelium-lined, thin-walled channels that collect fluid from the tissue spaces and return it to the blood. Lymphatic vascular system consists of endothelium-lined thin-walled channels that collect fluid from the tissue spaces and return it to the blood. This fluid is called lymph. This fluid is called lymph, and it circulates in only one direction, toward the heart. This fluid is called lymph, and it circulates in only one direction, toward the heart. There are three types of lymph vessels, and they can be distinguished based on their size and morphology. And they based on their size and morphology. The smallest one, lymphatic capillaries. Next, lymphatic collecting vessels. And the biggest, the largest, lymphatic ducts. Lymphatic ducts. Lymphatic capillaries. Lymphatic capillaries begin as blind-ending tubes in connective tissue. Lymphatic capillaries begin as blind-ending tubes in connective tissue. In connective tissue. The basal lamina is absent, and the endothelial cells do not form tight junctions. The basal lamina is absent and the endothelial cells do not form tight junctions, which facilitates the entry of liquids into the lymph capillary, which facilitate the entry of liquids into the lymph capillary. Temporary openings in the endothelial lining of the lymph capillaries also allow the entry of larger particles into the lymph capillaries. Temporary openings in the endothelial lining of the lymph capillaries also allow the entry of larger particles into the lymph capillaries. Collagen filaments and coring filaments link the endothelium to the surrounding tissue, prevent collapse of the lymphatic lumen. Prevent the collapse of the lymphatic lumens. Next type, lymphatic collecting vessels. Lymphatic collecting vessels. They are formed in the result of the fusion of some lymphatic capillaries. You can see lymphatic collecting vessels formed as a result of fusion of some lymphatic capillaries. The lymphatic vessels form walls. Lymphatic vessels form walls. The lymph is moved by the compression of the lymph vessels by surrounding tissues. The lymph is moved by the compression of the lymph vessels by surrounding tissues. The direction of lymph flow is determined by the walls. The direction of lymph flow is determined by the walls. Lymph vessels empty intermittently into lymph nodes. Lymph vessels empty intermittently into lymph nodes, from which the lymph continues in efferent lymph vessels. In efferent lymph vessels. And the last type, lymphatic ducts. Lymphatic ducts. Lymphatic ducts. Lymphatic ducts contain one or two layers of smooth muscle cells in their wall. They also form walls. They also form valves. Peristaltic contractions of the smooth muscle contribute to the movement of lymph towards the heart, in addition to the compression of the ducts by surrounding tissues. Peristaltic contractions of the smooth muscle contribute to the movement of lymph toward the heart, in addition to the compression of the ducts by surrounding tissues. The heart structure. The heart structure. The heart is a muscular organ that contracts rhythmically, pumping the blood through the circulatory system. The heart is a muscular organ that contracts rhythmically, pumping the blood through the circulatory system. It is also responsible for producing hormone called arterial natriuretic factor. Arterial natriuretic factor. The heart wall consists of 3 tunics. The heart wall consists of 3 tunics. First one, endocardium. Endocardium. Endocardium consists of endothelial cells, subendothelial layer of loose connective tissue, muscular elastic layer, and external connective tissue layer. And external connective tissue layer. Between the endocardium and the myocardium is a connective tissue subendocardial layer, which consists of veins, nerves, and branches of Purkinje cells. You can see here branches of the Purkinje cells. Next layer, myocardium. Myocardium. Myocardium is the thickest and consists of cardiac muscle cells. The thickest layer, which consists of cardiac muscle cells. And third outer layer is epicardium. Epicardium. Epicardium is the serous covering of the heart. Serous covering of the heart, forming visceral layer of the pericardium. It is covered by simple squamous epithelium, mesothelium. Look to the picture. 3.5. Mesothelium layer. And contains loose connective tissue. Loose connective tissue, which can contain blood vessels, arteries and veins, and nerves. Here you can see slides of the heart. Endocardium layer. Endocardium layer consists of endothelium, smooth, simple, squamous epithelium, subendothelial layers, very thin here, muscular elastic layer, and external connective tissue layer. And you can see Purkinje fibers between endothelium and endocardium and myocardium. And myocardium is very thick and consists of different cardiac muscle cells. On another picture we can see endocardium, myocardium, and epicardium. There are three types of the cardiac muscle cells which we can find in myocardium. There are three types of cardiac muscle cells which we can find in myocardium. First type, contractile cardiac cells. Contractile cardiac cells, or ordinary or working cells. They form the basic part of a myocardium and are characterized highly developed contractile system. They form the basic part of a myocardium and are characterized highly developed contractile system. Next type, conductive cardiac cells. Conductive cardiac cells. They have ability to generation and conduction of electrical impulses through an impulse generating and impulse conducting system of heart. Conductive cardiac cells have ability to generation and conduction of electrical impulses through an impulse generating and impulse conducted system of heart. And there are two types of conductive cardiac cells. Pacemaker or nodal or arrhythmic cells or P cells are the modified cardiac muscle cells with pale cytoplasm and fewer myofilaments. These cells are situated in the sinoatrial and atrioventricular nodes and internodal tracts. And next type, Purkinje or conduction fiber cells, form atrioventricular bundle of the heart. Purkinje fibers are modified cardiac muscle cells found in the subendocardium of the ventricles. Subendocardium of the ventricles. And they constitute part of the specialized impulse conducted system, which connects to the right and left bundle branches and regulates the heartbeat. Regulates the heartbeat. These are large muscular cells with a vacuolated cytoplasm. We are speaking about Purkinje fiber cells. These are large muscular cells with a vacuolated cytoplasm due to the high glycogen contents due to the high glycogen content. Other characteristics that help distinguish Purkinje fibers from typical cardiac muscle cells are that they contain fewer myofibrils and more sarcoplasm. They contain fewer myofibrils and more sarcoplasm. The bundles of Purkinje cells travel in the subendocardial layer to the apex of the heart. The bundles of Purkinje cells travel in the subendocardial layer to the apex of the heart, where they begin giving off side branches that make contact with working cells. Where they've been given off side branches that make contacts with working cells. And the last type of cardiac muscle cells in myocardium is secretory cell. Secretory or endocrine cardiac cells. Secretory or endocrine cardiac cells are found in arterial cells and are characterized by weak development of contractile system. In their sarcoplasm, near to poles of a nucleus, there are granules containing hormone. In their sarcoplasm, near to poles of a nucleus, there are granules containing hormone. Arterial natriuretic factor, auriculin, and arteriopeptin. Auriculin or arteriopeptin. And let's look to subendocardium layer when we can see Purkinje fibers vacuolated with pale cytoplasm. Purkinje fibers between endocardium layer and myocardium layer. And here we can see a lot of cardiac muscle cells in myocardium. Development of the heart and blood vessels. Development of the heart and blood vessels. We can distinguish 5 stages to heart development. We can distinguish 5 stages to heart development. First, specification of cardiac precursor cells. Specification of cardiac precursor cells. Second, migration of cardiac precursor cells and fusion of the primordia. Migration of cardiac precursor cells and fusion of the primordia. Next stage is heart looping. Next stage, heart chamber formation. And the last stage is septation and valve formation. septation, and valve formation. Development of the heart here. The primordium of the heart forms in the cardiogenic plate, the part of the splaghnopleuric mesoderm, located at the cranial end of the embryo. The primordium of the heart forms in the cardiogenic plate, located at the cranial end of the embryo. Cardiogenic area, cardiogenic plate. Angiogenic cells clusters, which lie in a horseshoe shape configuration in the plate, coalesce to form two endocardial tubes. Two endocardial tubes. Cells cluster lie in horseshoe shape configuration in the plate, form cardial tubes. These tubes are then forced into the thoracic region due to cephalic and lateral folding, where they fuse together, forming a single endocardial tube. Fusing together and form single endocardium heart tube. The tube can be subdivided into primordial heart chambers, starting caudally in the inflow end. The sinus venosus, primitive atria, ventricle, and bulbus cordis. So we can distinguish primitive atrium, primitive ventricle, bulbus cordis, and truncus arterioles. Next, the heart tube begins to grow rapidly, forcing it to bend upon itself. The result is the bulboventricular loop. Bulboventricular loop. You can see here. Bulboventricular loop. Septa begin to grow in the atria, ventricle, and bulbus cordis to form right and left atria, right and left ventricles, and two great vessels, the pulmonary artery and the aorta. Septa begin to grow in the atria, ventricle, and bulbus cordis. You can see septa begin to grow in atria, ventricle, and bulbus cordis, and form right and left atria, right and left ventricles, and two great vessels, the pulmonary artery and the aorta. By the end of the 8 weeks, parturition is completed and the fetal heart has formed. By the end of the 8-week, parturition is completed and the fetal heart has formed. The lesson is over. Thank you for your attention.